Hello students, now we are going to discuss about the impurities in pharmaceutical substance, right? So when we talk about the pharmaceutical substance, these are nothing but medicines or any formulations, right? In that formulations there may be the active pharmaceutical ingredient, the active drug. Apart from that, there will be the excipient, binding agent, adherent, or if it is suspension, suspending agent, if it is the emulsion, emulsifying agent, or so many excipients, those which will also be there, right? So along with that, there will be the some types of unwanted chemical which actually it is not required into that pharmaceutical substance, but even though that will be there, right? And the presence of those unwanted chemicals, it will directly or indirectly affect to the therapeutic value or therapeutic property of that particular pharmaceuticals, right? So basically let us try to understand what impurities are actually, right? So the topic is the impurities in pharmaceutical substances, right? So let us try to understand impurities in pharmaceuticals are unwanted chemicals. That's what unwanted, which undesired, which is actually supposed to not be there, but it is there because of so many reasons that we are going to discuss the sources of impurities, right? Unwanted chemicals that remains with, right, that will be remains with the active pharmaceutical ingredient, API, whatever the active pharmaceutical ingredient which we are using to formulate any pharmaceutical product, right? So along with that, it may be there, it may be remains, right? Then developed during stability testing. So many stability testing we are doing for the pharmaceutical formulation, right? So during stability testing, it's physical property, chemical property, biological property. When we are trying to check its solubility, it's what you call optical rotation, refractive index, if it's different values, the chemical, how it is lone pair of electron, it is releasing or it is withdrawing, what kind of things whenever we are trying to get, it's how long it can be stable during storage. If you are testing those drug, that time also there is chances that impurity may remains over there or may develop over there. Coming to the third reason, developed during formulations. Whenever we are doing manufacturing, whenever we are formulating any tablet, capsule, ointment, lotion, syrup, spirit, whatever it is, the formulations, ophthalmic, parenteral, right? During that process also it may develop, right? Coming to the next region, upon aging of API and formulated API in medicines, right? As soon as the time pass, or the particular active pharmaceutical ingredient, when it will reach to its maximum times or time limit, it will cross. Also the long time storage, it will also cause the developing of impurities onto those, right? So that's what in short what we can see, the presence of these unwanted chemicals even in small quantity or small amounts may influence or may affect the quality, the purity and the efficacy. Ultimately the goal is of the any pharmaceutical formulation that it should be quality one, then only we can expect the good health, and it should be safety. First of all, it should not produce any side action or side effect, adverse reaction. It should it should show the proper therapeutic property or therapeutic efficacy. That's what efficacy, what for it is meant for. It is for anti-diabetic, it is for anti-inflammatory, antipyretic, analgesic, what kind of pharmaceutical product of the medicine it is manufactured for. In those proceedings, those kinds of effect when we are expecting from any patient body, that should be proper. It should not affect or interfere because of the presence of impurities. If they are there in the small quantity also, definitely they are going to interfere in that proceedings. Coming to the definition, according to the ICH, International Council for Harmonization, this is one of the governing body, right, which will regulate the different types of toxicity study and the rules and regulations. It is international body where the 29 countries are the member in that body. So this particular agency, it has defined the impurities. So according to the ICH, how we can define it, any component of medicinal products, right? Any component which is there in the medicinal products, which is not the chemical entity, right? Which is not the chemical entity defined as active substance or an excipient in the product. So those are impurities, right? So what is happening here, the presence of these impurities in pharmaceutical should be controlled and monitored, right? It has to be controlled. It should be, it can be there, we cannot avoid these things to be there in the pharmaceutical, but there is a limit. That's what limit test it is there. In coming classes we are going to discuss the limit test for chloride, phosphate, heavy metal, arsenic and all those impurities which will be developing keep on during the manufacturing of the pharmaceutical product, the storage condition. There are so many reasons that also we are going to discuss, right? So that's what the as of now with this class we can able to understand that the impurities is something which is unwanted, undesired. It should not be there along with the pharmaceutical product, but even though they may develop, they may be remains over there, and ultimately if they are there, definitely we are going to compromise with the quality, safety, purity and the efficacy. So that's what it is important to know, that what is the percentage, what is the whether they are in below the limit or not, that we have to understand, we need to determine as a pharmacist, as a quality control department, right? So that's what impurity in pharmaceutical products it becomes very important, because these are something very serious concern which is not supposed to be there with the pharmaceutical drug. Thank you.